we have changed our name from the Leetown Science Center to the Eastern Ecological Science Center as we combined with Patuxent. So I just want to put that in. Next slide, please. So as Greg said, I have talked over a number of years, mainly presenting some of the data that we collected during a long-term monitoring and assessment study that was a collaborative project with USGS and state agencies in Pennsylvania, West Virginia, and Maryland. We collected samples from 2013 through 2019, and we're still analyzing and trying to summarize a lot of that data. What this was basically integrated monitoring of surface water chemical concentrations, where we looked at well over 300 different chemicals, included comprehensive fish health assessments of adult smallmouth bass in both the spring, pre-spawn, and the fall when they would be going through recrudescence in reproduction, and then health monitoring of young of the year in the early summer. Surface water samples were collected monthly and twice a month in the spring with some stormwater samples. When this started, it was primarily focused on the endocrine disruption and intersects that we had observed, and we were seeing that more associated with agricultural land use. So we really focused on pesticides, hormones, phytoestrogens, with some other things like pharmaceuticals and wastewater associated chemicals more sporadically analyzed. We had 4 sites, 2 in the Potomac and 2 in the Susquehanna River drainages. Next slide, please. And we looked at really a lot of biological endpoints, things from, morphometric, age, condition factor, looking at visible abnormalities. We collected blood samples. We used some of the plasma for vitellogenin and then archived other plasma samples. We collected tissues for all organs for pathology. We collected samples of liver and anterior kidney. Anterior kidney is similar to our bone marrow. It is the immune function producing organ of fish. So we basically have sets of over 100 different genes we're looking at. And then in the later 3 years, Cheyenne Smith, a graduate student from WVU, we collected anterior kidney for immune function assays. And then we also archived tissue for hoping we'd get, throughout the time, funding for chemical analysis. In the young of the year, most of those fish were very small, so they were collected and visible abnormalities were documented, and then they were fixed for pathology. Next slide, please. The 4 sites that we ended up with varied in land use, and in addition to all the biological and chemical data, we have also collected a huge amount of land use data, things like pesticide application in the various watersheds. So we've looked at both the immediate catchment, which is the hydrologic unit containing the sampling site, and then also what we call the upstream catchment scale, which captures things that are going on upstream of the site. The 4 sites are Pine Creek and West Branch Macantango Creek in the Susquehanna drainage, and then Antietam Creek and the South Branch Potomac site near Moorfield. Next slide, please. And these do have very different land use. You can see that in the second column, percent agriculture, Antietam Creek 49%, West Branch Macantango 32%. So those are our high ag sites. South Branch has 14% and Pine Creek 9%. We also wanted to differentiate between pasture and cropland. So you can see again, Antietam Creek has about half pasture, half crop. This is in the upstream catchment, whereas South Branch Pine have more pasture than crop, and West Branch Macantango has more crop than pasture. Next slide, please. What this has allowed us to do is really look at key exposure periods throughout the fish reproductive cycle, as well as health indicators. So for instance, in April and May is when we tend to see the fish starting to build nests. Their gonads are going through final maturation. In late May and June is when they typically spawn. So you have the eggs, and then you have the young of the year, which is really the time, a really sensitive time for developmental, both of reproductive as well as the immune response. And then later in the fall is when the adults start going through recrudescence. So those germ cells that are in the gonads begin to develop for the next year. Next slide, please. So just some of the things that over the years that we have found, as I said, we looked at well over 100 different pesticides and herbicides, and 3 of them really came up as commonly detected, and those are atrazine, simazine, and metolachlor. And what I have here, this is just the data from 2014 through 2017, showing the total number. So at all sites, we had well over 40 samples that we looked at in that approximately 3-year period. And you can see that atrazine and at West Branch and Antietam are almost always detected. And then in parentheses, after the total detections is the number of detections that are above the 100 nanogram per liter assessment threshold. So again, very common to see atrazine at fairly high levels. Simazine, we're seeing often, not at as often at the above the assessment threshold, and metolachlor similarly. Next slide, please. So this is just an example of some of the things we saw early on. So in 2015, this is a site comparison. And what you can see here is that for the South Branch and for pine, most of the detects are well below that 100 nanogram per liter assessment. But at sometimes, particularly at Antietam and West Branch Macantango, we have some very high levels. So for instance, in mid-April in 2015 at Antietam, high level 670 nanograms per liter. But if you look at the light blue bars, that's West Branch Macantango. And what you can see there is that atrazine levels were high and stayed pretty high through that late May, early into June level. And again, remembering that this is the time when fish that spawn in the spring are often going through really sensitive developmental times. You can also see in the West Branch that we had another peak in the fall. Next slide, please. We also, in addition to seasonal variations, we also see annual variations. So this is an example just of Antietam showing you 2015 through 2017. Again, we're seeing some very high peaks at certain times. If you look at 2016, you can see really high levels of atrazine, simazine, and metolachlor. Again, in that May into June period. You can compare that to 2017 when even atrazine and metolachlor were rarely above the 100 nanogram per liter assessment. Next slide, please. So, you know, early on, what that showed us is that fish and other aquatic organisms are being exposed to these complex mixtures of the parent compounds that vary seasonally and annually. And to really get a handle on what's going on, we need to be able to look at those mixtures and understand both. Sometimes there'll be synergistic, sometimes antagonistic effects. And that's what we're really trying to tease out with some of this. But the other thing that really we began to be aware of is, first of all, some of the other compounds that can be in there, either as active ingredients, or as Dr. Bennett has already given you a good background for, contamination in some of these pesticides. And PFAS is something that certainly has in the last few years raised a lot of concern. And then the other issue is some of the metabolites, degradates, or transformation products of the parent compounds that can be occurring in aquatic systems. Next slide, please. So in 2018 and 2019, a different analytical schedule was used for pesticides, and this measured a number of the metabolites. For atrazine, there were 5 metabolites that were measured. The one in italics, the dialkyl atrazine, is the one that we saw the highest concentrations of, and I'm going to talk a little bit about, but I will say that the other 4 were pretty commonly measured as well, although at lower concentrations. For metolachlor, 4 metabolites were measured. The first 3 we rarely found, and they were at low concentrations, but the last one, the metolachlor, ethane sulfonic acid, we did find at fairly high concentrations. Next slide, please. So here's an example again. I'm just picking out some of the data. This is Antietam. The red line is the metabolite. The blue line is the parent compound of atrazine. And again, showing 2 years, 2018 and 2019. And again, you see there's some really different annual patterns. In 2018, parent compound was fairly low, similar to the 2017 that I showed in the previous slide. But in 2019, again, we had some very high peaks of the parent compound. Next slide, please. One of the interesting things when we compared, did some site comparisons, is the difference sort of in these metabolites of the parent compound. So if you look at Antietam and the atrazine levels, and West Branch Macantango and the atrazine, the parent compounds, they're not that much different. So if you look at, for instance, May 8th, 174 nanograms per liter at Antietam and 176 at West Branch Macantango. So fairly similar as far as the parent compound. But in this case, when you look at the metabolite, you can see that Antietam, we throughout the year had very high levels of that metabolite. West Branch Macantango, the levels were much lower and were sometimes non-detectable. Next slide, please. With metolachlor, again, you see a somewhat similar pattern in that the metabolite is significantly higher at most sampling times than the parent compound. Again, in 2019, we saw some peaks similar to the atrazine peaks. Next slide. In this case, when we compare the 2 sites, we see sort of the opposite of what we saw with atrazine, where at Antietam, the levels of the metabolite are lower. I mean, they're still quite high, but they're lower than what we were seeing at West Branch Macantango. So there's a lot we still need to learn about what is it that's regulating these metabolites, how long they're going to stick around, and even more, what effects do they have on the aquatic organisms. Next slide, please. So some of the takeaways to date are that we definitely need a better understanding of the effects of mixtures, exposure to mixtures at key developmental periods. We also need to understand the environmental factors that regulate the breakdown and transformation of these parent compounds. And then really there's so little known on the effects of these pesticide metabolites. There have been a few studies that have shown they often can be as toxic, and in some cases have been reported to be more toxic than the parent compound. Depends on what organisms you're looking at and what endpoint you're looking at, but definitely something we want to pursue. Next slide, please. Now I want to just quickly change, add a little to what Dr. Bennett already discussed as far as the PFAS. When this became an issue, as I mentioned, because we tend to archive as much tissue as we can, hoping that funding may come up and we may be able to do further studies, we had archived plasma samples from all these fish. And so we used the 2018 plasma samples and had 13 different PFAS compounds measured in the plasma. These were analyzed by a commercial company up in British Columbia, SGS Axis. And I just want to point out that the concentrations are in nanograms per ml, which is parts per billion. If you remember, Dr. Bennett was talking about parts per trillion. So these are significantly higher levels. Next slide, please. And what we found was that of those 13, 4 compounds were detected in every single plasma sample, even Pine Creek, which is sort of our forested reference site. The PFOS, which, as Dr. Bennett mentioned, is an 8-carbon molecule, was the most commonly found and at the highest concentrations. But 3 others, also high carbon numbers, the longer chain carbons, were found. And then 2 additional compounds were detected in some samples. But what you note here is that all of them are the longer chain carbon. And these are the ones that have been shown to bioaccumulate and biomagnify through the food chain. And smallmouth bass are one of the top predators as far as fish go. So not surprising that they have somewhat high levels. Next slide, please. We found that there were definitely site differences. So for the PFOS, every single site was significantly different, with Antietam having the highest concentrations, West Branch Macantongo the next highest, South Branch third, and then Pine Creek having the lowest. When you look at the other 3 compounds that were found in every fish, the pattern is the same, but South Branch Potomac and Pine Creek were not significantly different than each other. So they were lower than West Branch Macantongo, which was also significantly lower than Antietam. Next slide, please. Based on these initial findings, we then went back and looked at samples from 2017 and 2019, so we would have a temporal comparison. We also looked at quite a few more sites to look at a spatial distribution. As far as Antietam, you can see that there seems to be a slight increase. They're not significantly different in both the total PFAS and the PFOS. Next slide, please. When you compare this with other studies, it's a little difficult because many of the studies are analyzing PFAS in muscle or liver. Muscle, because as is mentioned, one of the human exposure routes is certainly through food. And so muscle is what people eat. And so the states, as far as setting consumption advisories, are looking at muscle. And most species that have been studied have found that the blood and the plasma have higher levels than the liver, which tends to have higher levels than the muscle. So I do want to point out that these are plasma. We wanted to look at the plasma because our interest is primarily on the fish health. And obviously what's circulating is getting into all organs and exposing cells. And I will say that worldwide, when you look at studies that have looked at blood and plasma levels, our results at Antietam are some of the highest. There was a study that was higher, and that was Cape Fear downstream of an actual PFAS production plant, where the striped bass had higher levels. Next slide. So conclusions, we do feel that the PFAS concentrations likely to have adverse effects are detected at some of the sites in the Chesapeake Bay watershed. Actually, Swatara Creek up in Pennsylvania, when we looked at that, had higher levels than Antietam. And we have done some correlations with various biological responses that we're seeing, and we are seeing correlations. Similarly, we know pesticide application may be a source, but there are many other sources that we have to consider. Wastewater treatment effluent, both from domestic and industrial sites, runoff and groundwater contamination from military facilities, airports, other places that use the firefighting foam, landfill leachate, and then biosolid applications is another major source. So we need to do a lot more research on sources to determine if pesticides are a major contributor. A more recent analytical schedule that we're using for additional samples is looking at 40 different of the PFAS compounds rather than just 13. And so that will give us an ability, I hope, to start teasing out sources. As I mentioned, we do see correlations with a number of our endpoints. And what we really now need to do is go into the lab using the concentrations that we have measured in the field and do some environmentally relevant exposures in the lab, both for PFAS and the pesticide transformation products. And those are the things we'd like to be doing in the next year. I will just mention one. Dr. Bennett mentioned about PFAS and potential looking at vaccines and whether you have a good response to the COVID vaccine. One of the interesting things that we did see as a correlation was that lymphocyte function in the fish was negatively correlated with some of the PFAS concentrations, and lymphocytes are the cells that produce antibodies, are one of the cells, one of the things that lymphocytes do. So there is a potential that we are seeing some of these same effects in fish as well. Next slide, please. And I would just like to acknowledge that, as I mentioned, this was funded through the USGS Ecosystems Mission Area, and we got much help from many of the state biologists in Pennsylvania Fish and Boat Commission, Pennsylvania Department of Environmental Protection, Maryland Department of Natural Resources, and West Virginia Division of Natural Resources, and much help with students.